So, we have been discussing about the various aspects of nucleic acid structure, how the whole thing started with fiber diffraction, single crystal X-ray data on shorter oligonucleotides has helped in obtaining great details about the structure of the nucleic acids at atomic resolution. The various aspects of the structure we have discussed, and we mentioned about the different types of models, the bDNA, aDNA, cDNA, ddNA and things like that. We also indicated the importance of this nucleic acid with regard to the hereditary functions, and we also talked about one of the new discoveries which is called as the zDNA, and here actually this slide is a continuation of that discussion and makes a direct comparison of the most prominent nucleic acid structure, which is the bDNA, and then we have the left handed which is the zDNA, and this is the comparison here of the two. By and large you can see here this bDNA, which is the one which is the prominent one in all systems. It is a right handed helix. You can see the helix goes in this manner, and there is a duplex. Of course the other strand also goes in this manner here, both are right handed helices, but they run in opposite directions. So, the 5 prime and to 3 prime end one sense, other one goes from the other end 5 prime end to 3 prime end, and they are held together by the base pairs here, and these base pairs are called the Watson-Crick base pairs. So, these are all the base pairs, and it is the overall features of the bDNA are given here. So, you can see there is a big groove which is indicated by this here. This is the big groove, it is called the major groove, and there is a smaller groove which is called the minor groove here, and therefore you can see that all the base pairs, one end opens in the minor groove, the other end opens on the major groove. So, these are the characteristic appearances of the bDNA. On the other hand, the zDNA is actually a left handed helix. You see the helix goes like this, and it is also zigzag. It is not as smooth as it is as the bDNA is. It also goes in a zigzag manner, and you have the left handed helix as indicated by this, okay, and there is a deep groove here, and there is a bulge at this point. So, therefore, this structure is not as smooth and symmetric as this one is. And here are the parameters which are given for these two structure types. This is the firstly right handed DNA, bDNA is right handed versus a left handed DNA here, and the number of nucleotides per turn. So, when the system repeats itself, the cycle repeats itself, how many nucleotide base pairs are there? The bDNA has 10 base pairs, and therefore we say 3.4 angstroms into 10, there is a 34 angstroms is the rise per turn, that is indicated here next. And then whereas the zDNA has 12 base pairs per turn, okay, and the diameter of this helix, the entire helix, here this is 20 angstroms and here it is 18 angstroms, somewhat less. And the rise per base pair is 3.4 angstroms here, this is 3.7 angstroms here, and the total helix pitch, that is 34 angstroms, because if you take 10 base pairs per turn, 3.4 into 10, that makes it 34, whereas this one goes to 45 angstroms, one particular turn. What is the rise? What is the rise per one turn? 45 angstroms here. And the random per base pair, there is a rotation per base pair, this is 36 degrees and this is minus 30. What is minus 30 degrees? Because it has to go in the negative sense, because it is a left handed DNA. If I take the right handed rotation as positive, the left handed rotation will be negative, and therefore it is minus 30, because 36 into 10 will be 360 degrees, that makes a complete turn whole way, and minus 30 into 12 makes it 360 degrees, and therefore that is a 12 into this will be minus 30. And there are grooves here, okay, wide and shallow, 3 different kinds of grooves, and these are very well stacked, and the base pairs are not very well stacked in the zDNA. So, you see the stacking is not very good, whereas here the base pairs are parallel to each other and they stack one over the other, and that is quite a good structure, symmetrical structure. And now there is one more important parameter which is not of course visible from here, that is called as the glycosidic torsion angle. What is the glycosidic torsion angle? I will show you that soon. And then I also mentioned in the last time that the in the case of bDNA it is a monomer which is the repeating unit. Monomer is the repeating unit, whereas in the zDNA it is the dimer which is a repeating unit, and that is why these are given separately here. And for the bDNA there are two kinds of glycosidic torsion for the zDNA. Two kinds of torsion angles are indicated here, syn and nt. One of the nucleotides, the guanine nucleotide, has the syn conformation, whereas the cytidine nucleotide has the nt conformation. And the sugar geometries, whereas in the bDNA they are all C2 prime endo, which is uniform, whereas here it alternates. So C3 prime endo guanosine has the C3 prime endo sugar geometry, and the cytosine has the C2 prime endo sugar geometry. Therefore this makes the dinucleotide as the repeating unit. Now what is the glycosidic torsion angle? This is explicitly indicated here. So you see here, this is the sugar ring. The sugar ring is a 5-membered ring here. This is shown for the RNA, but it does not matter, it is the same for the DNA as well. So in the case of DNA, this OH group is replaced by the hydrogen, same here. Okay. Now the glycosidic torsion angle is this. This is the glycosidic torsion angle, rotation around this bond. Okay, this is labeled as chi. Okay. And a proper model of that, this is the schematic on the left hand side, and the proper model of that is shown on the right hand side. So here it is all the things are included in this. So this torsion angle, you notice here what is the difference between these two. Here it is shown for the purine ring, this is a purine base, okay, and this one is the guanine. What is chosen here is the guanine because you have an NH2 group here, and adenine also has an NH2 group here. So this is guanine has an NH2 group here, adenine has an NH2 group there. Okay. So if the rotation is around this bond, what is the difference between these two? See, the 5-membered ring, this is the 5-membered ring of this purine ring, comes on the side of the oxygen here. This torsion angle is defined with respect to these 4 atoms. Okay, if you can either define this way or you can define with respect to these 4 atoms. So either way it is the same thing. So you can choose a convention where you want, how you want to choose, one particular convention is used. So the essential point to note is, from the structure point of view, is that the 5-membered ring here comes on side of the oxygen, and here there is a proton attached. Notice the proton is not shown here, but there is a proton here. There is a proton here, and that proton is called as the H8 proton. Okay. And in this case this proton is here. Okay. So the protons are on two opposite sides in the two cases. Now in this case this is called as the syn conformation, whereas this one goes outside compared to relative to this oxygen position, and in this case it comes closer, and this is called as the NT conformation. Typically it is shown in terms of the structure. When you actually build a model, how does it look? So you have the C2 prime endo geometry here, sugar ring is in the C2 prime endo geometry, and the anti-glycosidic torsion angle. So this orients rotation around this bond is indicated here. You see this NH2 groups comes close, and this is the proton, this is the indicated, this is the proton here. You see this proton comes closer to the oxygen of the sugar ring here. So whereas that proton is far away, this far away compared to the oxygen, that is this one here, this is far away as I mentioned. This is far away, but this one will now be, this will be closer to the sugar ring protons here, the H1 prime protons, that will be close. This proton will be close to the sugar ring protons here, whereas this proton will be farther away from the H1 prime or the H2 double prime protons in the case of the NT conformation. C2 prime endo sugar and the NT position of the glycosidic torsion angle. So, and these will be important. Why I am mentioning this is because this will be extremely important when we actually use these information for structure calculations with regard to determination of the structures, what are the short distances from the NMR point of view, which are the important distances one has to see. From that point of view this is very important. That is why I am pointing out these things, a definition of the glycosidic torsion angle. So we have C3 prime endo geometry for the sugar ring in A type structures or RNA structures, and B type structures it is the C2 prime endo sugar geometry, and the glycosidic torsion angle is NT conformation. It is both in bDNA and aDNA, whereas you have possibilities of glycosidic torsion angle in the syn and anti conformations in the case of zDNA, and there are other possibilities as well. Now okay, is that all about the nucleic acid structure? Surely this is not. Why we are saying this? Because you see, you remember the DNA, if it were a duplex as indicated here for so long, the length of the DNA will be of the order of centimeters. Okay. Considering 1 billion base pairs and DNA length in this every cell, it will be in the order of centimeters, because 34 angstroms per rise and 1 billion base pairs if you take, it will come to the order of centimeters. Now this cannot be accommodated in a cell which is only of 1 micron size, and the nucleus is even smaller than that. Therefore DNA folds multiple times. There may be stretches of various other kinds of strands where other kinds of base pairing possibilities, there may be single strand possibilities, all sorts of things can be present. And in the case of RNA, we mentioned already that the mRNA is a single stranded RNA, and in the case of tRNA and ribosomal RNAs, there can be different kinds of structures possible with various kinds of folds of the backbone and resulting in various different kinds of base-base interactions. What are the possible base-base interactions? Okay, that is what we are going to see here. So you can have, so far we talked about the purine pyrimidine base pairs, that is the one which is indicated here on the top, AT base pair. Okay, now this is purine pyrimidine base pairs. So what is, this is the pyrimidine ring and this is the purine ring, this is AU and GC. For the AU, how many hydrogen bonds are there? 2 hydrogen bonds, and for the GC or for the GC we have 3 hydrogen bonds which are holding them together. Okay. Notice carefully what is the nature of the hydrogen bond. This particular position is hydrogen bonded to this nitrogen here. Okay. And what is R? R is a place where the sugar ring is attached. Here the sugar ring is attached in this position, and in this case in the purine sugar ring is attached at this position. This position is called as the N9 position. This is N9 position and this is N1 position. This is N1, the nomenclature goes in that manner. Whereas for the pyrimidine, this position is called as the N3 position. N3, the numbering goes in that manner. Okay. So the standard nomenclature with regard to the IUPAC conventions, etc., you have, this is the N9 position, this is the N1 position, this is the N3 position. This is all we have to remember here. So far as the purine pyrimidine base pairs are concerned, these are the Watson-Crick base pairs which are there in this bDNA. So duplex, the backbone is in green and the purple are the base pairs. This is the possibility. Now there is another possibility also here. See, look what is involved here. So what has happened here? This base pair, this is different from this base pair, the reverse Watson-Crick. This portion is the same. Okay, the A is in the same configuration, NH. Now what has changed in this? NH, NHN, the R is here, whereas the R is here. In this case the R is up, which means there is a rotation with respect to that, with respect to this axis, there is a 180 degrees rotation. Therefore this R has come on to the top, this R is at the same place. Okay, the sugar ring is at the same place. Here the sugar ring goes on the top. Okay, so therefore this is called as reverse Watson-Crick, and same happens here as well. Okay, NR is here and in this case the NR is there. So this is both in the AU as well as the GC base pairs, it goes in this manner. Now, so this is the other possibility of hydrogen bonding. Surely these people had toyed around with all these possibilities, finally came up with this. It turned out that this, what they came out was finally the correct one with regard to the bDNA. But this kind of things also people have toyed around to see whether this one fits. Some experimental data did the building model building did not fit into this one, so they came out with this. But this can be there, in other kinds of situations where the DNA folds and various kinds of interactions can happen, these ones can be there. And then look at this is called as the reverse, this is the AU Hookstein, this is the Hookstein base pairing. Hookstein base pairing, what happens here? The NH is base pair to this, this configuration is the same here. Okay. Now this is the purine ring and it is this nitrogen It is this nitrogen, the 5 membered ring nitrogen is involved in the hydrogen bonding with this NH here, okay. Whereas in this case it is the NH of the N of the 6 membered ring, whereas here is the N of the 5 membered ring. So that is here, this is free here, right, this N is free, and that now comes here to take part in the. So therefore that is a rotation of the glycostatic torsion angle which brings this nitrogen closer to this hydrogen here. This is the N3 of U is based to N7 of the purine, and that is called as the Hookstein base pairing. So Hookstein probably is the one who proposed this. Similarly there is the reverse Hookstein base pair. In this case the NR here, NR is here, NR is there on the same side of the duplex or on the base pair, that is they are both open in the same groove, whereas here you see the NR is here and this NR is here, and therefore this is the AU reverse Hookstein base pair, okay. Once again this is the N7 N3, but the glycostatic torsion angle is different, and therefore this one has gone on to the other side, U reverse Hookstein, and this one is a wobble base pair. Now then you have the so called wobble, well, so that was the reverse Hookstein. Now this is the wobble base pair, wobble base pair GU wobble. See, so far we talked about the AU base pair, but here it is a GU now. The GU is not the thing which is normally supposed to happen, we have to have a GC base pair, okay. But here we are now talking about GU base pair, the G pairs with U. Conventionally we are AT base pair or AU base pair, but here it is the G pairs with U, this also is possible. And now this hydrogen bonding scheme here is like this. So this N1 of G pairs with this oxygen here, and the once again this is the normal GU base pair, okay, NR here, and the reverse is again the NR goes on the other side. So it depends on which oxygen is involved in the hydrogen bonding, whether it is this oxygen or this oxygen. That is what determines whether it is in the normal way or it is in the reverse way. So both this is called as the wobble base pair, because this is normally not there in the duplex DNA. And then similarly you have the AC, you see here this is the AC reverse Hookstein, okay, AC reverse wobble and AC reverse Hookstein. AC reverse meaning what, we normally have AT base pair, right. So when the T is replaced by C we call it as a wobble. AC is a wobble base pair, but this is also possible. So such hydrogen bondings are also possible. So you have this NHN here, NHN, both are NHN hydrogen bonds in this case. Okay, in all of these cases one is these are NHO hydrogen bonds in this case. In these ones you have NHN hydrogen bonds, one NHO hydrogen bond. In all of these one NHN, one NHO, here also one NHN and one NHO, whereas here you see AC in this GU wobble both hydrogen bonds are NHO, and the reverse also both are NHO hydrogen bonds, and in this AC reverse they are both NHN hydrogen bonds. NHN hydrogen bonds is the amino proton is participating in the hydrogen bonds. Therefore you see how many different possibilities are there for hydrogen bond formation. Now then you can also home your purine base pairs. Just as we talked about purine pyrimidine base pairs, you can also have purine base pairs. So here it is A and AA base pair, okay, and different types. So AA N1, okay, N1 to amino. So you have the one of the proton is the amino and the other one is the nitrogen, okay. So you have here this amino of this is hydrogen bonded to this nitrogen here of the 6 membered ring, and similarly 6 membered ring of this is paired to this nitrogen here A, okay. This is one AA N1 amino symmetric base pair, and then you have AA N7. N7 is involved. In this case you see it is the N7 which is hydrogen bond to the amino. This is the N7. N7 is hydrogen bond to the amino NH proton here, and correspondingly for this one N7 is paired to the hydrogen bond here. So this is we have the amino protons participating in this, and similarly here as well AA N1 amino, N7 amino. So here you use both N7, both places N7, here we use both places N1, and here we use N1 and N7. One case it is N7, other case it is N1. So it is enormous possibilities of hydrogen bonding schemes are possible, and of course only when, of course you cannot remember this, but certainly when you actually have to build models, try and understand the structures which you actually observe, then one should take into account all of these possibilities. How does one determine all of this? This of course one determines by NMR, by looking at which are the ones which are hydrogen bonded, which are the protons which are involved in the hydrogen bond, whether it is aminoprotons, which nitrogen is involved. So all of this can be determined by from NMR data. So this is the G1 N1 carbonyl symmetric, CG, GG. So you have G1 is this, okay. Now G1 is hydrogen bonded to the carbonyl here. Earlier you are using NN hydrogen bonds. So here is NHO. So NHO hydrogen bond here, okay. N1 position of this goes to the oxygen of this, N1 position of this one goes to the oxygen of this, and both here in this case only the 6 membered ring is involved. And what happens here? Now this is GG N3 amino symmetric, okay. Now here it is NHN hydrogen bonds, and this is the amino to N3 position, this is the N3 position. In this case this was used N1 position, here we are using the N3 position and N3 to this amino proton, okay. GG N3 amino symmetric hydrogen bond. And the last one is GG N7 N1 carbonyl amino. So N7 that is this one, this is the NHN hydrogen bond here to the N1, and the amino is hydrogen bonded to the oxygen here. So you have here one NHO hydrogen bond and one NHN hydrogen bond. Similarly GG N1 carbonyl N7 hydrogen bond. So N1 is where? This is the N1, N1 is bonded to the oxygen here, this is N1 is go to the oxygen here, and this N7 is going to the amino. So this is the N1, there is the amino proton on the G, this G amino is hydrogen bonded to the oxygen, whereas this nitrogen is hydrogen bonded to the amino of the G. So these are GG base pairs. So you can have so many different kinds of base pairs, homeopurine base pairs, we saw AA base pairs, and then we have the GG base pairs, different possibilities of hydrogen bonds. Then heteropurine base pairing. So far we looked at AA or GG, but you can also have GA base pairing. So GA base pairing is indicated here, different possibilities of GA pairing here. So the G here you see N1 carbonyl amino. So it is the amino of the A is hydrogen bonded to the carbonyl of the G, and the amino of G is hydrogen bonded to the nitrogen of A. So this is NHN hydrogen bond and this is the NHO hydrogen bond. So this was GA N1 carbonyl amino, okay. So these are both N1 positions there, N1, okay, and these are GA, this is A. Notice here in the case of G there is a proton here, I indicated to you earlier, okay, and what is present in the case of A? There is a proton at in the case of G there is an amino here, there is an H2 group, this is position number 2, at 2 position there is the amino group here. In the case of A at position number 2 there is also a proton, okay, and you have an amino group at this position there, okay. So this is the proton here is N2 position, this is N9 and this is 8. So there is a H8 proton here and the H2 proton here, okay. Let me write that here, this is H8 and this is H2 and this is H8. So these are the positions according to the labeling, and that is what we have there, okay. So in the same manner you have the GA N3 amino N1, GA N3. So where is G? This is the G and N3 is this, and this is going to the amino of the A. So this is the NH2. So this NHN hydrogen bond is there, and this amino, this is at the 2 position, is hydrogen bonded to the N3 of this, okay. And so therefore you can NHN, these are both NHN hydrogen bonds, here one NHO and one NHN, and involves amino groups and the iminoprotons. And the next one is AgN7N1 amino carbon. So this is the GA one possibility, and this is the next possibility here. So this amino is going to the oxygen of the A amino is going to the oxygen of the G. E amino is going to the oxygen of the G, okay, and then this NHN, and this is the N1 position is going to the N7 here, N1 to N7 AG, okay, N7 N1, this is A, this is N7, this is N1, and AG N7 amino N3. And look at these possibilities, because there are so many hydrogen bond acceptors and donors in the base structure. All these acceptors and donors can accept a hydrogen bond and give a hydrogen bond, give a proton, and that is why you have this so many different kinds of possibilities of hydrogen bonds. Okay. Pyrimidine base pairing as well. Likewise, so far we looked at the pyrimidine base pairs. You have possibilities of CC symmetric base pairing, CC carbonyl amino symmetric base pairing, and Cu, there is a Cu pairing possibility although N3 here, and then UU pairing carbonyl N3 to symmetric, and we will not describe it in detail further. So just the main structures are shown here, okay. Then you have possibility of UC N3 and UU hydrogen bonding possible. So you have so many different possibilities of hydrogen bonding, and because all of these nitrogens and the carbonyl oxygens, they are all acceptors, and you have the donors are the amino protons, the NH2s and the NH protons, and these are present on both all the bases. You have such kind of acceptors and donors, and that is why you get different kinds of hydrogen bonds. Okay. Now as a result of all of these, now I indicated here what are the different possibilities of the structures. We said so far we talked about the various kinds of base pairing schemes, and where do they occur? Where do they occur? Okay, now you have possibilities of a duplex here, standard duplex. Then you can have, when a running chain is going on, depending upon the nature of the sequence which is present here, it can assume different kinds of secondary structures. Single stranded regions, it has a single stranded regions, and you have a loop here, okay. There are loops coming out here in this place, okay, and these are the, so single stranded regions you will have in this is not paired at all. So this portion is paired, this is not paired, so all kinds of folding schemes can occur, okay. So these are, and then you can have the hairpins. So the thing go like this looks like a hairpin, right. So therefore, and then you have these bulges here. In a duplex in between, the base sequence is such that it is not pairing possibility here. This fellow bulges out, so you will have a bulge here, okay. So similarly, there is a small bulge here, there is only one, but this is several bases are involved in this, therefore it becomes a longer loop, and this is only one base which is going out here. Such kind of possibilities are there. And now in the internal loops within the duplex itself, you can have certain regions where there are bulges in the middle. So because these are not complementary, why does it happen? Because these are not complementary sequences. When there are not complementary sequences, the possibility of a base pair does not exist, then you will have these kinds of loops, okay, symmetric loops in the parallel structures, and you can have antisymmetric structures, antisymmetric loops, and you have these kinds of possibilities, and you can have 4 stranded structures. So in this place, so you have the 3 stem, this structure has 3 stem, this structure has 3 stem, okay, and we will see that such structures do occur in RNA, ribosomal RNA, tRNA and things like that, and there is an actually a region which is bulged out here. So the chain has to run like this. So these are all the possibilities indicating the folds of the nucleic acid structure, and you see here is a 4 stem structure, and such kind of structures do occur in the functional aspects. Whenever there is a kind of a recombinant process going on inside in the replication process or such kind of structures do happen in this. This is a 4 stem process. So the stable DNA structure, RNA structure is one thing, but during the when there are activities going on, the DNA has to open up and interact with other systems, other DNA segments or other protein segments, and such kind of transient structures do happen. And these transient structures, for example, one of them is called the holiday junction. So you have here, this is the 4 stranded structure. So there are many such kind of structures which are possible. These are functionally relevant. So the DNA has to open itself to express itself to form either for producing the proteins or replication process and things like that, and in all those processes such kind of transient structures do occur. Okay. Now so far we talked about the 2 bases interacting with each other, but you can also have base triples. Base triple means, so you have 3 base pairs, 3 bases are interacting with one another. See, here you have the normal Watson-Crick AT base pair, normal Watson-Crick AT base pair, and here the normal Watson-Crick GC base pair, and on the top comes here a third base which is the T. So you have a TAT triplet, this is called as the base triple, TAT triple, and here it is a CGC triple. Okay. And look here, in this base pairing what happens, in the free position which is here, the N7 position is used up for this base pairing in the for the third base. Okay, the oxygen of this uses this amino proton, this proton here, okay, amino proton, and the N7 is used up for the T and this place. So this kind of this triple base pairing is possible. Similarly for the GC. GC also same thing happens, the free donors and the acceptors in the Watson-Crick base pair can accommodate another base in the major groove. If it comes on this other side it is a major groove, so on this side it is a minor groove. So you can have this sort of possibilities here. So you have a GC and the G coming here. Okay, what we had here, TAT, T is the pyrimidine, AT, and then of course the third base is the pyrimidine. Here there is a third base which is the purine, the G is coming, and similarly in this case you have a AT pair and then the A is coming, the third base is A. And now what are these arrows here? These arrows are indicating which are the short distances, because these are the observables in NMR spectra. See, these what are indicated are the proton-proton distances. So all these proton-proton distances are the ones which are short distances, and we use these kind of short distances to identify or assign the individual bases and the individual protons. That is why these are indicated here, indicated by double headed arrows, that we can actually observe these ones by recording spectra in water. Now these are base triples. Now you can also have quadruples, okay, it is called the G tetrad. So 4 G's can hydrogen bond with each other and to form what is called as the GGGG tetrad. There are 4 G's here, very symmetric. See, and all the donor and the acceptor sites are used up here. So it uses this nitrogen, this oxygen, this nitrogen iminoproton and this aminoproton. So all of these are involved in the hydrogen bonding in a very symmetrical manner. So this produces a G tetrad, which is an extremely stable structure, very stable structure. We will see, I will show you examples of this, how this 4-stranded structure is possible and how this can be extremely stable. Okay. So typically how does the base DNA triple helix look like? So you have the green one is a normal duplex, and the triple helix comes, the third strand comes in this major groove. Okay, it comes on this, and then you see the third strand is hydrogen bonded to the base pairs in the duplex, and this is the space filling model of the same sort of a structure. And these ones are such kind of structures are possible in the quadruplexes. So the 4-stranded structures are possible here. So you have the G tetrad, and you can have quadruplexes of various types of quadruplexes are possible. These are called parallel stranded quadruplexes, and here you have 2 strands going in one direction, 2 strands going in other direction, and you can also have loops here. Okay, the chain runs like this and loops around come here, the other chain runs like this and loops around and comes down here, and these 4 G's which are in the thing, they can form hydrogen bonded structures. And in all of these is the glycosidic torsion angles becomes important. Okay. So and depending upon what is the orientation of this, you can have different kinds of base pairing schemes, and so that determines the base pair structure. Then the last one structure which has been discovered some time back is called the I motif. The I motif is a structure which is between CC plus. See, the CC base pairing is possible, but the one of the C gets protonated here, and because of that protonation it gets a positive charge, therefore it is called as the CC structure. Now here what happens is you have 2 duplexes interdigitating, that is why it is called as I motif. Okay. So it can be different molecules, or the same molecule loops around, turns around and things like that and comes back, and that is shown is in this case the chain starts here, let us say, goes like this, and then goes like this and then turns around, and then So, these are the various possibilities, that all these are required, because the DNA has to fold in many different ways and all those structures will have to be stabilized. This stabilization happens because of the different possibilities of hydrogen bonding pairs. So, this is actually a quadruplex. How does the quadruplex structure look like? So, this shows here the how are the 4 strands accommodated in this, okay. So, that is the quadruplex structure. You have these 4 colors here, these are the individual strands which are going, as you can form a structure of this type. This is the DNA quadruplex. So, I think we possibly stop here, and now of course here the DNA protein complexes can be formed. DNA protein complexes are important in various. Now here it shows how a duplex DNA can fold and accommodate the proteins, and the proteins can interact with the DNA, and proteins will have to express and protein will have to interact at various sites in the DNA duplex or with the other various kinds of structures that are possible, either with the single stranded areas, the double stranded areas, different possibilities of interactions are possible. So there is a whole variety of structural possibilities, enormous complexities in the DNA structure, and people thought the only duplex DNA is a simple thing, that is the only thing about the DNA. But of late we know that there is so much more about the nucleic acid structure which remains to be explored. Much more needs to be done with regard to the nucleic acids and especially RNA structures. So many different varieties of RNA structures are possible. So I think with that I will stop here.